Hi everyone, welcome back to my Surat video tutorials. Today I'm going to show you some important Surat visualization functions. So Surat have some important plot functions to enable us produce beautiful figures for publication or presentation. So in my previous videos, I already showed you the dim plot, spatial dim plot, feature plot functions. Today I'm going to go over dim plot, feature plot, violin plot, dot plot, ridge plot, and do heat map function to show you these functions in more details. Then you can generate beautiful images. So I'm going to use the Surat object created in video 17. In this tutorial, we integrated single-cell RNA sequencing data between healthy controls and fibrotic human lung. So we can read the data in. Okay, first, let's use the dim plot function again to see the cell clusters. Just to put a Surat object name here, then we can run dim plot. Let's zoom in. So you can see we have 4 types of cells in the lung: epithelial cells, endothelial cells, mesenchymal cells, and immune cells. You can see with the default function, cells are not enabled. So now we can put a label on. Let's run this one. You can see now cell clusters are labeled with mesenchymal cells, endothelial cells, immune cells, and epithelial cells. So if you prefer the dark background, we can use the dark theme feature to change the background into dark color. If we run, we can zoom in. You can see now the background of the plot is black. So if you don't want the legend on the right hand side, we can use the feature no legend. Let's run again. You can see now the legend is gone. We can also change the size of label for UMAP1 and UMAP2 for the x-axis and the y-axis. Let's run this one. You can see now we have much bigger letters for UMAP1 and UMAP2. You also can add a title on top of the figure. We can use the feature labels title. Here I put the title as the control and the IPF. Let's run. You can see now we have a title on the top of the figure. We can zoom in. You can see title is here. So in my previous video, I already showed you how to choose your favorite color for each cell cluster. You can also change the point size for cells and the label size for the letters. You can also put a repel tool if you have lots of clusters to label, or you use longer names for each cell cluster to avoid the letters overlapping. So we can run this one. You can see the difference. Now you can see the color changed for the cell clusters, and also the labeling size for the letters are changed. So for the spatial dim plot function, I introduced this function in my video 8 and video 9. So today I'm not going to show it again. If you want to watch it, you can go back to watch it, my video 8 and video 9. So another important plot function we frequently use is the feature plot. We use this function to see the gene expression in different cell clusters. For example, we frequently use the feature plot with marked genes to identify the cell types in different cell clusters. So we can run this feature plot function with marked genes EpCAM, CLDA5, collagen 1, A2, and PTPRC. So we have the feature plot in this window. You can see down here there's a warning. Surat could not find the PTPRC in the default searching locations, but it found PTPRC in RNA-seq. So this is because we perform the data integration and we set the default assay as integrated. If we set this back to RNA, so now we run this feature plot function again. So you can see down here we don't have that warning anymore. So we perform the integration between control samples and IPF none. Let's use the view function to have a look at the metadata. So you can see in the metadata, the column of origin ident, we have control for healthy donors and IPF for fibrotic nuns. So let's go back to the R scripts. So now we can use the submit by feature to compare a gene expression in control and fibrotic nun. For example, we know CTHRC1 is a gene that is upregulated in fibrotic nun in fibroblasts. So we can run feature plot for CTHRC1 with the submit by function. We can zoom in. You can see now we have the feature plot for CTHRC1. So now you can see we can compare CTHRC1 expression in control samples and IPF samples. You can see CTHRC1 has a higher expression in the mesenchymal cell population. We know CTHRC1 is upregulated in myofibroblasts. For the feature plot, we can also use the minimum cutoff function to reduce the background. Here I'm going to use the num as the example. Num is a gene expressed in fibroblast. So we can use the feature plot function to generate plot 1 without cutoff. Then we generate plot 2 with the minimum cutoff 1. Now we can see plot 1 and plot 2 side by side. Let's run. We can zoom in. You can see in plot 1 we have the expression level from 0 to 5. You can see some background level in other cell clusters, but in plot 2 the expression level is from 1 to 5. and the background color is gone in other cell canisters. So we know this cell canister is the canister for fibroblast. Using this feature plot function, we can also see co-expression of some genes in the cell canisters. What we need to do, just put this feature blend equals true in the code. Let's run it now. We can zoom in again. You can see now we have different colors. Here is the color pattern. We know both DCN and NAM are expressed in fibroblast. So you can see DCN expression in red color, NAM expression in green color. If there is a co-expression, then the color changes to yellow. So this is a very similar figure like we do immunohistochemistry for protein markers staining. Now I'm going to introduce the function of wireline plot, and we frequently use it to plot the M features, N count, and percentage of mitochondria DNA to perform quality control when we do the analysis. So if we run the wireline plot now, we can see the N features, N count, and the percentage of mitochondria DNA in each cell canister. Also, we can use the wireline plot function to see the gene expression levels. Once again, we are using the 4 mark genes. Let's zoom in. So you can see the wireline plot for APCAM. It is highly expressed in epithelial cells. CLDN5 is highly expressed in endothelial cells. Collagen 1A2, we use it as a mark gene for mesenchymal cells, and PTPRC is a mark gene for immune cells. Also, we can use the wireline plot to compare gene expression in control and fibrotic lung samples. We knew collagen 1A1 is highly upregulated in fibrotic lung, and also CTHRC1 is upregulated in myofibroblast. So we can compare the expression for both genes between control and fibrotic samples. If we zoom in, you can see, so left hand side are control and the right hand side are fibrotic. You can see collagen 1A1 has higher expression in the fibrotic cell canisters. The same for CTHRC1. It has higher expression in the endothelial cells, mesenchymal cells, and immune cells. With the wireline plot function, we can also plot the row count for different genes. What we need to do is to set the slot in count, or you can set log2 to change the x-axis. If we run and zoom in, you can see the original count for 4 mark genes. Each gene marks a different cell canister. So now we are going to show the dot plot function. This function is frequently used to generate figures for publication as well. Because dot plot can show lots of gene expression pattern on the same figure. So here I used 2 genes for each cell type. EPCAM and FTPB for epithelial cells, CLDN5 and PCAM1 for endothelial cells, collagen 1A1 and LAM for mesenchymal cells, PTPR for immune cells. Let's run the dot plot. We can zoom in. You can see the big blue dot. The blue color shows the expression level, and the dot size shows the percentage of cells express this gene in the cell cluster. So the dot plot can show gene expression in a cell cluster by the expression level and the percentage of cells in a cell cluster that express the gene. Now I can show you another plot function called a Regge plot. Again, we are going to use 4 marker genes. Often you don't see this plot in publications because the images will use a lot of space. Let's run the Regge plot function for 4 markers. We can zoom in again. You can see for the Regge plot, PTPRC is expressed in the immune cells, collagen 1A1 is expressed in mesenchymal cells, CLDN5 is the mark for endothelial cells, and EPCAM is the mark for epithelial cells. So in this video tutorial, the last plot function I want to show you is the do-hit map function. Similar to the dot plot function, do-hit map is frequently used to show mark genes for the cell clusters. So we can use find all markers function to find the marks for each cell cluster in our dataset. We set this feature as true to return positive markers and with a minimum percentage expression 25% in each cell cluster, and the fold changes threshold as 0.25. So let's run this function to find the markers for each cell cluster. We know we enabled 4 cell clusters in our dataset. Let's run. So you can see down here, Surat is calculating mark genes first for the epithelial cells cluster. The next is for the endothelial cells. Now Surat is calculating mark genes for the cell clusters of mesenchymal cells. Finally, is the cell cluster of immune cells. So after finish the calculating, Surat will generate an object we named as control IPF combined markers. So you can see here we have this data now. We can press the data, have a look. You can see the genes for epithelial cells, p-value, fold changes, the percentage of expression in epithelial cells, and the percentage of expression in other cell types. So we can go back to the R scripts. Now we can group the genes by cell clusters and also arrange according to their fold changes, then show top 5 genes for each cluster. So you can see here we have data for top 5 genes for each cell types, because we have 4 types of cells, then in total we have 20 genes. So now we can use the do-hit map to generate a heat map for top 5 genes in each cell types. Because we are using the integrated dataset, we set the default say as RNA. To hit map function requires default say as integrated. We need to switch back the default say as integrated for running the doHitMap function. So let's switch it back to integrated. Then we can run the doHitMap function. Now we can zoom in. You can see we generated a heatmap for top 5 genes for each cell type. Okay, I'm going to stop here for today's demonstration. Please remember to subscribe my channel. I'm looking forward to see you again in my next video tutorial.